Good morning, everyone. It's my pleasure to introduce Dr. Deepak Srivastava to Stanford. Dr. Srivastava is the president of the Gladstone Institute at San Francisco, director of the Center for Stem Cell Biology, professor in pediatric developmental cardiology at UCSF, and a pediatric cardiologist, to name a few. Dr. Srivastava completed his medical training at the University of Texas, residency at UCSF, fellowship at Boston Children's, and followed the Physician Scientist Development Program with Eric Olson. Dr. Srivastava has single-handedly revolutionized the field of cardiac genetics and stem cell reprogramming to understand the developmental basis of congenital heart disease. Almost everything we know today about the genetics of congenital heart disease is from Dr. Srivastava's work and his team's work. For these contributions to the scientific community, he has received numerous awards, including being elected to the National Academy of Sciences, the American Heart Association Distinguished Scientist Award, the Upstart 50 Inventor Award, and many, many more. He's on the editorial board of numerous top-tier scientific journals, has published over 200 papers, has mentored dozens of scientists and physician scientists, many of whom have gone on to have successful research careers. Today, Dr. Srivastava will discuss the genetics and environmental basis of birth defects. Thank you for being here.
Thank you, Shishma. It's wonderful to be back here, and I have so many friends that I get to see here. And I was just thinking, I was actually a fourth-year medical student, spending a month in pediatric endocrinology here 35 years ago, I think in 1989. And the place has changed quite dramatically since then. And I congratulate you all on what a wonderful clinical and scientific effort you have here today. Let me see if I can, how do I get to my first slide? There we go. What I thought I'd do today is share with you all some of the excitement that is going on in science right now. And I'd say for most human diseases at the moment, we finally have an opportunity to understand the bases for why these diseases occur at a depth that we've never had before, largely because of advances in a number of tools and technologies, including AI that you hear about all the time. And specifically for pediatrics, and specifically for birth defects, is where I want to focus my comments. And about, as most of you know, about 5% of all children worldwide are going to be born with the birth defect. And by and large, in the past, we've simply had the opportunity to describe what happens and study the phenomenology. But we're finally in a position where we can begin to understand exactly why those defects occur. And that's a prerequisite to being able to develop either ways to prevent those defects from occurring in the first place, which would be the ideal, or in most cases, accepting that they've occurred, but then finding ways to mitigate the ongoing sequelae of those defects. And so to do that, what we really have to understand is exactly which cell types in the body or in an organ that's been affected are in play that results in those birth defects. And it's really only very recent technology that allows us to go from an organ level analysis to an actual individual cell level analysis. And then as we understand mutations that are present in people who have various birth defects, we now have the ability to not only say this cell is affected, but a mutation affects this cell in this exact way, ultimately hoping to make that an actionable information. And I won't say too much about this today, but as we've learned the developmental biology of these individual cells, it actually affords an opportunity to utilize that developmental biology knowledge in the adult for regenerative purposes, because essentially then you can say if nature was using a certain pathway to make a cell during development, well, maybe we could reuse that in the adult to make whatever cell type we want. Now, because I am a pediatric cardiologist, I'll focus my comments today on the most common of all birth defects, which of course is congenital heart disease, affecting over 1% of all children born worldwide. But what I want to impress with you is that the principles that I'll share with you on how we go about figuring this out are really applicable to every birth defect. So whichever organ system you're interested in or taking care of, I think just please think about it, the principles which will apply to whichever area you're studying. And I'll also say that one of the sequelae of at least congenital heart disease has been quite difficult for us to deal with. And that is, as we've gotten better and better at keeping children with complex congenital heart disease alive, what we're developing is a group of folks that reach later years in life, including adulthood. And as they age, a number of consequences have been arising, including heart failure, which we think of as an adult problem. But with the now 1.5 million survivors of congenital heart disease just in the US, many of these children, as they get older, are suffering from heart failure. So that is going to be an increasingly large problem to solve. Now, one of the most severe forms of congenital heart disease is shown here in this slide, where children can be born with either just a single right ventricle or a single left ventricle. And these are obviously very severe conditions. We can keep these children alive, and now these are the children who are reaching adulthood and often suffer from heart failure, as I mentioned. But what this slide also illustrates is that even in conditions where a whole chamber is missing, the rest of the heart is there and pretty normal. And what that would suggest even years ago is that to us is that there must be separable molecular pathways that are governing formation of each of these regions of the heart relatively independently to one another. And over the years, as we began to understand the developmental biology of the heart, it became clear why that might occur. And that is that we now know that there are independent progenitor cells that are coming from different parts of the body that go on to make the different chambers. And so I've tried to schematize that in this slide here, where I've color-coded various different progenitors in the heart. And this is now a relatively crude diagram. And for each of these colors that you see here, we now know actually multiple different cell types that make them up. But to simplify, we can trace back that very early in development, even after 2 weeks after fertilization, we start to parse different cells in the developing embryo that form this crescent shape that you see here. And these red cells will go on to ultimately form the left ventricle of the heart. These yellow cells will go on to form the right ventricle of the heart, and many other cell types, as you see here, color-coded. But what this suggests is that you can imagine that you might have mutations that affect, say, just the yellow cells. Well, that could be a good reason why you might then now be missing the right ventricle. You might have mutations that just affect the red cells. Well, that would be a reasonable way to think about how you might be missing just the left ventricle. And so now that we know this, and we know subtypes of cells in each of these colors, we can start to say that a mutation might affect just a small number of cells, even within the yellow or red cells or blue cells. And that might be the basis of heart disease. And now we're getting very deep into understanding at that level. And so my laboratory for some years has been focused on deeply dissecting the gene networks that are controlling every cell type that is contributing to the heart that I just showed you, with the hope that that would allow us then to understand, as we understand genetic mutations, the molecular basis for why these defects are occurring. And also, as I mentioned, that we might be able to actually create new muscle cells. And I won't have time to talk about it today, but I'll say a little bit during my Heart Center grand rounds at noon about how we've been able to superimpose some of these developmental networks on heart cells in the heart that are not muscle cells and get them to change their fate and become new muscle for regenerative purposes. Now, I mentioned earlier that we have the technology to not go just study the whole organ like we used to, but study individual cells in the organ. And that has really been a revolution in single cell biology. And many folks here at Stanford have been pioneers in this. And this really allows us to now take, instead of looking at the whole heart, disperse the heart into tens of thousands of cells, individual cells, and study every one of them in a single experiment. And that, for the first time, allows us to study not just how a mutation would affect an organ, but how mutations would affect individual cells. And that's the basis of a lot of what we've been able to understand lately. Now, what I'm going to focus most of my comments on today are the genetic side. And our laboratory has published a number of studies over the years understanding the genetics of congenital heart disease. And between the work from our lab and many, many other labs across the world, including the efforts of the Pediatric Cardiac Genomics Consortium that Dan Bernstein and I have jointly led a part of that project, as part of that consortium, we can now begin to explain nearly half of all congenital heart disease at a genetic level. That still leaves over half that is unexplained, that as a community we're still working on. But within these, there are a few big categories. There's the things that are environmental effects, there are aneuploidies, and then there are single gene mutations in the coding or non-coding regions. For the environmental effects, I'll just say a couple of words, because many of those situations, we may know that some environmental stimulus results in a congenital heart disease. And so we think, okay, now we understand that. But in fact, for most of them, we know that the phenomenon occurs, but we don't know exactly how. And again, that is, unless you know how, you can't really intervene in a thoughtful manner. And so our laboratory has recently tried to tackle this for the most common of all environmental insults that leads to an increased risk for congenital heart disease, and that is maternal diabetes. So about children who are born to mothers with diabetes, this is not gestational diabetes because that happens later, but who have diabetes at the beginning of pregnancy, have about a 5-fold higher increased risk for having a child with congenital heart disease. And so using some of these single cell technologies that I mentioned, we were recently able to actually figure out the precise cell type affected by maternal diabetes and exactly how. And as I mentioned, it's really this, going from this notion that we know that there's an increased risk to how. And very briefly, in work that was published last year, I'll just summarize for you schematically what we found at a conceptual level. And that is that in fetuses exposed to higher glucose levels, there is a change in the anterior-posterior signaling, patterning, if you will, of the embryo. And this anterior-posterior patterning is critical for development of most organs and is particularly true, for the heart, where there's a gradient of retinoic acid signaling that sets up an anterior-posterior gradient normally in these developing, particularly those yellow cells that I mentioned earlier. And in the setting of maternal hyperglycemia and fetal hyperglycemia, this is disrupted so that there's more posterior signaling, more retinoic acid signaling that extends to the anterior half of these progenitors, where it should just be in the posterior half. So conceptually, think of it just as a disruption of that anterior-posterior signaling. And so the cells are mispatterned and don't know exactly what they should be. And that we showed results in some of these birth defects. So it's getting to this level of granularity that is necessary. And the hope then is this could then lead to ways that even if somebody has diabetes before they're pregnant, we can learn enough about the mechanism that we can say, okay, you need to take this pill that would be preventive and lower that risk from 5-fold back down to normal. That's the ultimate goal. And now we're using our model to test such interventions. Now, what about aneuploidy? So this is the big chunk here. And that makes us feel good. Okay, we know that trisomy 21 or trisomy 18 causes heart defects. Well, that's great, makes us feel good. But in fact, there are a lot of genes on chromosome 21, on chromosome 18, on chromosome 13. And so it doesn't really help if you're just at that level of specificity. And it's been difficult for decades to go from a large chromosome region to knowing which gene or genes in a region that's duplicated is causing the heart defect. And the most common of all of those, of course, is Down syndrome or trisomy 21, where about half of the children who have trisomy 21 will have a congenital heart lesion. And there's a typical type of heart lesion that I'm sure most of you are familiar with, which is atrioventricular septal defects that is schematized here, where there's often free communication between all four chambers that commonly occurs in Down syndrome, and in fact occurs with a 1,000-fold increased incidence compared to the non-Down syndrome population. So it happens without trisomy 21, but it happens 1,000-fold more frequently. So this is clearly something about trisomy 21 is specifically making this type of defect happen. And we just have never had a handle on what that might be. But we know that there must be some dosage-sensitive genes on chromosome 21 that when present at 50% higher levels, not a lot more, 50% higher levels is resulting in this. I'm giving you a teaser for this, which I forgot. That I'm giving heart center grand rounds, and I didn't want to completely overlap these talks, but I recognize that not a lot of the same folks will be there, and I'm really excited about this, so I wanted to share it with you without telling you how we got there, which I will at noon. But basically, we took a very systematic approach that's only possible because of single cell technologies and machine learning technologies present today, where we systematically tested all of the potential candidate genes on chromosome 21, one by one in a large screen to come to candidate genes that might be the causative ones, found one that was a good candidate gene by studying stem cells in a dish, and then tested that in vivo in a mouse model where there is a mouse that's trisomic that has heart defects similar to this. And there you can actually, if you have a candidate gene, you can just cross the trisomic mouse with the mouse that's heterozygous for your candidate gene. And now you've lowered the number of copies of your candidate gene from 3 to 2 because you've removed one. And we did that for one of the genes, which is something that affects gene regulation called HMGN1. The name's not important, but if we do this sort of cross and then look at mice that now we've removed one copy of this candidate gene and we count the number of offspring that have septal defects, you can see here, this is the normal mouse, this is the trisomic mouse, and if we remove one copy of this candidate gene, we've now rescued the cardiac defects in this mouse. And so I'll share the details at noon today, but as a teaser, I think we've been able to use the technologies we have today to actually, for the first time, figure out a critical gene. And I should say the mouse genetics would suggest that it's probably 2 genes that together have to be increased in dose. And another group had shown a second gene called DYRK1A that is likely contributing with this. Okay, the last part of the talk, what I'd like to focus on is sort of single genes when mutated are sufficient, not increased risk, but sufficient to cause disease. And those are genes typically that have very, very large effect all by themselves. And so that's all you need. And I'd say most of congenital heart disease and probably most birth defects are what we like to call oligogenic, where it's likely a handful of genes that together, each with a smaller effect, come together. And actually Casey Gifford, who's here, when she was in my lab several years ago, actually probably made the first and only true experimental demonstration that really does occur in humans, and demonstrated that in an animal model. But those are complicated to study. And the story I want to share with you is about a monogenic disorder that's taught us actually quite a bit about the biology of a very common form of heart disease that involves the aortic valve. So it turns out that when we say congenital heart disease occurs in 1% of children, that's actually wrong. And it's a gross underestimation. It turns out that 1 to 2% of children born worldwide, have an aortic valve defect, a bicuspid aortic valve, that is a congenital defect. So really, it's double what I told you earlier, the incidence. And most of the kids who are born with this don't even know that they have it at birth. But as they get older, this birth defect has manifestations. And while there's a rare population that has critical aortic stenosis, where they need an intervention in the first few weeks of life with usually a catheter balloon dilation, most folks only develop calcification and stenosis later in life. About a third will get calcified as they get older, these valves, and then need either a surgical catheter mediated, open heart surgery mediated valve replacement, or more recently, a catheter mediated replacement. Now, this condition of aortic valve stenosis appears to be a spectrum, ranging from a child with bicuspid aortic valve who doesn't even know they have it until they're 60 years, to critical aortic stenosis where they need an intervention after birth, to hypoplastic left heart syndrome, where the aortic valve never even opens in utero, and they subsequently get the left ventricle doesn't even form, and they need a Fontan procedure. So this whole spectrum we've seen now in an individual family, which suggests that this is all part of the same disease, but along a spectrum. But we've never really understood the genetics of this or the mechanisms by which this occurs. And this is despite the fact that when people get to become adults, this calcification of the valve it turns out to be the third leading cause of adult heart disease, and of course is age dependent. And this looks something like this, where this is the bicuspid aortic valve that has 2 fused leaflets and over time gets calcified. So it was many years ago, now nearly over 20 years ago, that we came across this family. So actually, this is when I was, before I moved to Gladstone and UCSF, I was at UT Southwestern in Dallas. I took care of this little boy here down at the bottom of this family tree who had critical AS and needed a balloon dilation in the first 2 weeks of life. But when we took a family history, it was clear that there are many generations of folks that had aortic valve disease, and most of the rest of the folks in this family tree that are shown in black here actually had calcific aortic valve stenosis ranging from their 20s to their 50s. And they had all had open heart surgery. And so this is clearly autosomally dominant transmission and monogenic in origin. And so we were able, even back then, to map a single gene that was causing this. And this was work led by Vidhu Garg at that time, a fellow in the pediatric cardiology fellow with us, who now runs the heart center at Nationwide Children's Hospital in Columbus. But what Vidhu found at that time was that it was heterozygous loss of function mutations of a gene called Notch1 that was causing this disease with 100% penetrance. And Notch1 may or may not be familiar to you all, but it is a very, very well-studied protein that regulates genes, turns genes on and off throughout the genome. It's involved in development of most of our organs in the body. So it's curious that this family with just lowered the dose by 50% of Notch1 was having just disease in the heart and just in the aortic valve. But that is what the genetics were telling us. So this is great. It was the first time we had a genetic cause of this common form of disease. But then it was frustrating because mice, normally what we would do to understand mechanism, because just knowing this is nice to know, but doesn't really help us. What we need to understand is the mechanism by which it occurs, the disease occurs. And mice that were heterozygous for this gene were fine. And mice that were homozygous null died as early embryos because Notch is also important for the development of the whole vasculature because it's in all our endothelial cells. And so years went by and we really learned nothing. And we moved on and did other things. And then a few years later, Shinya Yamanaka, who had started a laboratory at Gladstone, also down the hall from us, had invented a revolutionary technology where we could take cells from the adult, at that time a skin fibroblast or biopsy, and now just a teaspoon of blood, and convert those adult cells, take them back in time and reprogram them now to be just like that own person's embryonic stem cells. And what that means is that you can then take those cells and turn them into anything you want. And here we were able to, we had other reasons to think that it was the lining of the valve, the endocardial lining of the valve, that was the culprit cell type. And so we could take these, what he called induced pluripotent stem cells, because we've induced them with the combination of 4 proteins to become what he called iPS cells, we could take those cells and turn them into endothelial cells. So we flew this family back up to now San Francisco from Dallas, and we made iPS cells from 4 people with the mutation and 4 people in the same family without it, thinking that that would sort of even out the individual variation between those, and we could figure that out. So we made endothelial cells. We did that. We differentiated them to endothelial cells. And then once again, we learned absolutely nothing. There was just too much noise in the system. Even though they were related, they had the difference in Notch1, a single base pair mutation, but they had millions of other differences as well. And so years again went by, and we really couldn't learn from this. And it wasn't until we had the ability to go into one of the patients with the Notch mutation with their iPS line and just switch out the one nucleotide that was mutated and correct it by gene editing, that then we could have the patient and a control cell line for the same patient that everything is the same except the one nucleotide in Notch1 that was mutated. And now we had what we call isogenic controls. And once we did that, then the biology laid itself out beautifully, and we were able to figure out the mechanism. And this required a large effort looking at a broad set of omics at that time, looking at where Notch1 binds to DNA and turns genes on and off, how it changes the epigenetic landscape, et cetera. And this work was led by a very talented MD PhD student, Christina Theodoris, who went on then, she was an MD PhD student at UCSF then. She went on to do a pediatric genetics residency at Children's in Boston. And after her postdoc, we've just recruited her to come back to Gladstone. And now she's become a generative AI expert and is training models with single cell data to figure these sorts of things out. But what Christina found at that time was that, I'll just schematize for you here in work we published years ago, is that Notch's normal job in the developing valve, is to stop a valve cell from becoming an osteoblast or bone-like cell. That's why it's there. That's its job. So you might ask, why would nature set it up that way? And it turns out that a valve, if you think about a valve, it has many similar properties as cartilage. And so nature has used the same gene networks to make a cardiac valve as it uses to make cartilage early on in development. But it doesn't want it to keep going and get ossified. So Notch is there to stop that process. So the disease is quite simple. It's when you lower the dose of Notch, it releases the brakes on this process, and these valve cells get confused and go on and start to change their fate. And they become more like an osteoblast cell, and they do what a good osteoblast should do, which is lay down calcium. And so the crux of this disease is really just a cellular reprogramming event where the valve cells gets reprogrammed to become more like an osteoblast cell. So that was interesting. But because of the way Christina went about figuring this out, she had mapped out the whole gene network that was involved in this cellular shift of a cell state. And it turns out that when she did that, she found that the upregulation of just 3 what we call transcription factors, which are, again, just like Notch, these proteins bind to DNA and turn lots of genes on and off. Upregulation of just these 3 that are shown here, the names aren't so important, what is sufficient for most of this reprogramming event. And that gave us hope that since the process funnels down to just these 3, that we might be able to screen for a drug that would prevent this cell fate transition. And we knew the whole network that was involved. So Christina set up a very clever screen to try to find a drug that just didn't just alter Notch levels, but the whole network, with the idea that even if somebody didn't have this disease because of a Notch mutation, they would likely still have the same cell fate switch as the basis. And so a drug we might find by screening for a network shift would apply to most people with the disease, not just those with Notch1. And so I won't go through the details with you, But what we had success in such a screen that again used a machine learning algorithm that Christina built at that time, and we found a drug that worked really beautifully in vitro, and this turns out to be an estrogen receptor related alpha antagonist. And so that was great. Now we had a potential drug. That was cool. But you'll recall I told you that the mouse that's heterozygous for Notch was fine. So we didn't really have a good animal model to test this in. And so some time went by. Again, I'm sharing you a 20-year arc of discovery. But Christina had another clever idea. And that is that I told you that this disease in humans is largely age dependent. And we know that telomeres get shorter as we get older. And we also knew that mice have 4 times longer telomeres than humans. So Christina asked, maybe mice are protected from the disease simply because their telomeres are longer. And that's a testable hypothesis because there's a genetic way where we can cross mice to get shorter telomeres. And so Christina did that experiment, and she made the telomeres a little bit shorter in mice. And lo and behold, the mice developed virtually the same phenotype as humans. It's really quite striking, and that's shown here, where this is a cross-section now through the aortic valve. And in the top panels, you see a normal valve, which you can see the nice 3 leaflets are thin. There are 3 of them. This is a high magnification. And down below are mice that are heterozygous for Notch and have shorter telomeres. And you can immediately appreciate this thickened aortic valve, just like occurs in humans. And by stained in red, which is a stain for calcium, heavily calcified. And it turns out we can look at markers of osteoblast in this, and they're positive for osteoblast markers also. So it looks like, this is there, and we have a good model now. And I want to come back to one point. I mentioned earlier that in humans, aortic valve disease appears to be a spectrum. So it turns out if we shorten these telomeres even further in mice, which we have a way to do by successive generations, they get shorter and shorter. The mice all died right after birth. They didn't even get to the stage of getting calcification. And when we dissected their hearts and looked at why they're dying because they basically had critical AS. They had critical aortic valve stenosis. And that's shown here. This is a newborn mouse that would have died. And you can see the aortic valve has a very tight opening. And by echo, we can do ultrasounds on these mice and show they have severe aortic valve stenosis. And so we think that this is, again, good evidence that this is really a spectrum of disease, and maybe in humans, that it's simply by virtue of their alterations in telomere length in different members of the family that we see that spectrum. So in any case, now we had a mouse model that it was calcified, but also was calcified enough that it caused aortic valve stenosis that could be measured by ultrasound. So that's what we do in patients. As you all know, if somebody has AS, we see it by ultrasound, an increase in velocity of blood flow across the valve, that then we can, that infers an increase in pressure gradient across that valve. And you can see here, these are mice with aortic valve increased velocity and pulmonary valve also in these mice after birth. And you can see it's incompletely penetrant, but about 15% of the offspring will have ultrasound AS that we can measure by ultrasound. So that's great. It gave us a model to test. So we tested the drug I mentioned to you in this first in a preventive fashion, asked, could this drug prevent disease? And in fact, it did a beautiful job of preventing both aortic valve and pulmonary valve stenosis. So that was great. But in humans, usually they were going to present with some degree of stenosis that we either auscultate or see by ultrasound. And then right now we just follow them over time. And when it gets bad enough, we try to make an intervention because there's no pharmacologic therapy for it at the moment. And so we did a different experiment where we identified those mice that had aortic valve stenosis and then took those and then randomized them into either placebo or drug. And then we came back and measured them aortic valve a month later to see how their valve stenosis progressed. Because just like in humans, this is a progressive disease in mice. And so what I'm showing you here is on the y-axis is the change in aortic valve peak pressure difference across the valve over that 1 month period in the placebo group. And you can see here, starting at baseline of, setting at 0, that most of these mice had an increase in aortic stenosis over that month, ranging anywhere from 2 to 3-fold to 50%, with an average of about an 80% progression over that time. And so in stark contrast, the mice that were treated with this estrogen receptor-related alpha inhibitor had a few mice that still had an increase, but the vast majority had very little to no increase in progression. And surprisingly, about half had a regression. So in this disease, we don't have to regress. If you just slow down the progression, particularly in the older population, they're going to actually have demise from something else. And so here we're very encouraged that this actually might even regress disease. And so we're very excited now that we might, for the first time, have a pharmacologic intervention for this very common disease that right now just simply requires surgical intervention. And the idea, hopefully, would be to have a daily pill that people could take that would slow down or arrest progression of this disease. And so we're now advancing this drug to a clinical trial first for those with the adults now with disease that is really a sequelae of this birth defect that I mentioned earlier. But I'm really excited about the possibility that we could actually prevent development of aortic stenosis in the fetus, even for this critical AS, and maybe even for hypoplastic, what the AS that results in hypoplastic left heart syndrome. And many of you know that we in cardiology have tried to put catheters across the fetal valve and blow it up, but these often restenose. But imagine doing that plus a drug that would prevent further thickening of the valve. That combination might have greater success. And so we're now trying to test in the mothers, this drug in the mothers of that, the model that I mentioned where they get critical AS as a newborn. And it'll be interesting to see if this actually works at that stage. So that story that I shared is probably, as a physician scientist, one of the most satisfying arcs of investigation of my career, because it's gone all the way from actually taking care of a patient while on service to figuring out the genetics underlying that, to the mechanism, and ultimately to a potential drug that could actually alter the natural history of this very common form of heart disease. So what I've tried to share with you today in the time we've had is how we can utilize this deep knowledge of developmental biology and how the system works, and use that either for regenerative purposes, and I'll say more about that in my talk later on today, but also to understand the human genetics of congenital heart disease. And I hope you could appreciate that most of what I indicated could be utilized for your favorite disease. And in fact, one of my colleagues at Gladstone, who's a neuroscientist, is using our similar approach for Down syndrome that I mentioned to see if that we can identify the genes involved in either the neurodevelopmental anomalies from a formation standpoint, or even from a cognitive standpoint and behavioral standpoint, because there, unlike some of the things for the heart, even after birth, I think we have the potential to intervene in a meaningful way to alter the natural history of the neurodeficits in Down syndrome. And others of our colleagues now are taking a similar approach for their favorite aneuploidy. And so I think the excitement in our field now that I want you guys to take away from this is that the tools and technologies that we have today to allow us to get at a depth of mechanism for disease that is unprecedented and is only going to increase from here. So for those of you who are training right now, either fellows, residents, medical students, your ability in your career to address your patient's needs is going to grow exponentially and it's the most exciting time in science and medicine ever. So with that, I'd like to thank the members of my laboratory, current members, a number of former members that I'm so proud of are running their own fabulous laboratory efforts, including Casey, as I mentioned here, at Stanford, and a number of great collaborators at Gladstone and beyond. And this is my current group at Gladstone. And with that, I'll stop and take some questions. Thank you very much.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.